Welcome to Chapter 3, Observing Microorganisms Through a Microscope. This chapter fulfills objective number 2, specifically microscope function and learning about the anatomy of prokaryotic and eukaryotic cells. In this chapter, we're going to go over units of measurement, specifically those that are used in microbiology, the instruments of microscopy, and preparation of specimens for light microscopes. In microbiology, we use the metric system. The metric system, once you get used to it, is actually easier than the English method because everything is based on 10. In microbiology, we specifically use the centimeter through the nanometer. Mainly what you need to remember is scale. A centimeter is what we use to measure very small objects, and millimeters are what we use to measure cells. Micrometers are what we use to measure things inside of a cell, and viruses, and nanometers are what we use to measure organic molecules. Let's look at these units of measurements and also what type of microscopes would be used to look at, things that are approximately those sizes. Now, up here we have a picture of a tick, ick, and we measure those in parts of a centimeter. They're very, very small, but you can still see them with your unaided eye. We can also use a light microscope like what you've been using in lab to be able to see them. Red blood cells cannot be seen with the naked eye. You have to look at them under a microscope. Now, they're in the range of 10 micrometers. You can see them with a light microscope. You can also see them with the electron microscopes. Prokaryotic cells, like bacteria, are significantly smaller than eukaryotic cells, like your red blood cells. Now, you can see that red blood cells are 5 micrometers. E. coli bacteria are 2 micrometers. And we can see them with a light microscope. This is getting about to the edge of what light microscopes you can easily see with a light microscope. Then to see viruses, you cannot see them with a light microscope. You can see them with electron microscopes. Actually, in this diagram, it shows that you can see some viruses with a light microscope, but they're very rare. Then if we want to look at DNA and other macromolecules, then we go to atomic force microscopes. Now on to microscopy. We're going to be discussing light microscopy, scanning acoustic microscopy, electron microscopy, scanned probe, and atomic force microscopy. Under the section of light microscopy, we're going to be discussing the path of light, why we use immersion oil, how to recognize micrographs that have been taken with light-filled, dark-filled, phase contrast, and fluorescent microscopes. Also, as you know from lab, you're going to need to know the parts of the microscope, so this diagram, this picture off to the side that's been labeled, would be of use to you, I'm sure. Light in a light microscope starts at the base in the illuminator, also known as a light bulb, and the rays of light go up through the condenser, which focuses the light, kind of straightens them out. Then it hits the specimen, and it shows the beams of light crossing here at the specimen, but what actually happens is it happens further up here. But the light is bent when it hits the specimen. Then when it hits the objective lenses, it's bent again, magnifying the image. Then we bounce back and forth through the prism so that instead of having to look straight down on top of the specimen, we can look at it at a more comfortable angle. Now I want you to keep in mind this path of light because we're going to be talking about it again, and especially when we talk about electron microscopes. Let's talk about why we need to use immersion oil when we use the highest magnification, the 1000x objective. Now, some of you may have experimented with this. You don't get real good focus with the 1000x without the immersion oil. So you're probably wondering why I have a picture of a fish. Well, the fish is not actually where you think it is when you look at it in the water. Light being bounced off of the fish bends when it goes from the water up into the air. It's because water is thicker than air. We call this having a different refractive index. Therefore, if you were to reach in and try to grab that fish, it wouldn't be where you think it is. Now, if the fish is in air, light doesn't get bent between the fish and your eyeball, and so it's where you actually think it is. We use this refractive indexes when we're looking at things under the 1000x objective. As light comes through, when it hits the glass, because the glass is thicker, it has a different refractive index than the air. It gets bent. Gets bent when it hits the air again. Here's another piece of glass, gets bent. Then when it hits a specimen and hits the air, some light waves can go off and not be funneled up into the objective. Now on the lower magnifications, that doesn't matter. There's plenty of light still getting from the specimen through the air into the objective. But once you get to higher magnification, you want all the light you can possibly get. That's why we add the immersion oil. The immersion oil has the same refractive index as glass, so light coming through the glass does not get bent, and more light ends up going into the 100x objective. For this next section, the book talks an awful lot about how the light waves get bent in different light microscopes. Other than the basics of what I've just gone over, I really don't expect you to know all of that for an introductory course. I do want you to be able to recognize different micrographs that have been produced by the different light microscopes. This particular micrograph was taken by what we call a bright field microscope. This is the type of microscope that we use in lab. It's a good workhorse variety of microscopes. They're inexpensive, they're useful in most applications, and they do the job. Light goes through and is blocked by the thicker portions of the sample showing up as dark spots. And it's a positive image, to use a photography term. Now, dark field microscopes, we'd mess around with how the light rays are bent, but we basically get a negative image of the organism. So you'll notice that the background is dark. That's why we call it a dark field microscope. And the thicker portions of the organism that are dark over here are actually lighter. Now you're wondering, why in the world would we want a negative image of an organism? Well, in doing this, the thicker areas pop out. Also, because most organisms are clear, they're kind of hard to see under a light microscope unless we stain them. If we stain them, the stains kill them. This way, we can look at organisms and they're light against a dark background, and it doesn't matter if they're clear. Also, we can look at living organisms. We don't have to stain them. We don't have to kill them. And some organisms are too fragile to be stained. It distorts them. Now, this next one looks very similar to the dark field, but you'll notice there's a little more color. There's a little more definition along the edges. This is a phase contrast micrograph. Once again, we start messing around with the light waves to provide contrast or shadowing. We do this for approximately the same reason that we use the dark field, to look at live organisms, to look at organisms without having to stain them if they're too fragile to be stained. Also, we get a better picture from a phase contrast microscope than we do from a dark field, but the dark field is less expensive. So it has a tendency to be used in situations where you wouldn't want to risk a very expensive phase contrast microscope. There are times when you want to stain just one thing in your sample. You want your stain to be very specific. So what we do is we take fluorescent molecules, fluorescent dyes. These are dyes that will fluoresce under UV light. You're familiar with it. You've all played with glow-in-the-dark toys. They fluoresce. You've also been to parties where they've had black lights and you laugh because your teeth and your white t-shirts fluoresce. Well, when we attach these dyes to an antibody, they can be very specific. Antibodies will attach to just the organism you're looking for. And you get a micrograph that looks like this. This micrograph was taken with a microscope that doesn't use light. It uses sound. It's kind of like sonar of the microbiology world. Sound waves are pulsed off of the sample, and a computer records how long it takes to come back, and we develop a 3D image. This type of microscopy is very useful for looking at living cells that are on top of something else. For example, it can be used to look at a bunch of microbes of biofilm that have been grown on a glass slide, or they can look at cancer cells on top of the organ if you're looking at a biopsy or something similar along that line. Now on to the electron microscopes. Light can only get us to a certain size. If you want to look at something smaller, you have to use electrons. It all has to do with wavelengths and different things that you don't need to worry about. Let's look at the transmission electron microscope. Now, it's like a light, bright-filled microscope that has been flipped upside down. We have the electron source up here at the top. Now, if you remember, with a light microscope, it was down here at the bottom, but the same principles apply. The source of the electrons beams the electrons down until it hits the sample, and actually the sample is right here, until it hits what's going to be looking at the electrons. Now, your eyes can't see electron beams, so we have a photographic plate that records that, and then we look at the photographic plate. Comes up on a nice big TV screen. Now, to focus the electron beams, instead of having glass lenses, we have electromagnetic condenser lenses. Now, electrons have what charge? That's right, a negative charge. So what charge would the condenser lenses have to have? That's right, a positive charge. So basically, we get electrical magnets that produce just positive charge, and we're able to focus the electrons, and we can get pretty darn cool pictures like this. Now here, because it's a transmission electron microscope, the beams of electrons are going through the specimen. The specimen has to be very thin, so we make thin sections of the specimen. The electrons go through, and we see the very small internal organelles of the cell. Now, electrons don't have color. That is a function of light that our eyes are able to perceive. So we take a computer and we put the color in because we like color. Now, if we want to look at the outside of an organism or an organelle on the inside, we use a scanning electron microscope. This one, the beam of electrons gets passed from the electron gun, it's focused by the magnets, and it's beamed across the surface of the specimen. And then you have an electron collector. It's like the photographic plate, and it goes up here to the TV screen where we see it. And you get lovely 3D images like that. This is actually the same organism, whereas this is a slice, we're looking at the outside of it. Okay, so that is the scanning electron microscope. Some things are too small for even an electron microscope to be able to magnify for us. So then we go to scanned probe or atomic force microscopy. This one is a scanned probe, this one's an atomic force. In both of them, a very, very sharp probe. Its point is down to one atom of metal, is either lightly drug across the molecule. Usually it's macromolecules we're looking at for this. Very lightly drug across the molecule, or is passed over the surface of the sample very, very close, and the flow of electrons from the sample to the end of the probe are measured. In this way, we can see very, very small things. Like in this situation, this is an enzyme that's used in DNA repair. This one's a toxin that's used by some microbes to punch through cell membranes. Now you're probably wondering what I want you to know from this section. What I want you to know is the path of light or electron beams. I want you to be able to recognize micrographs and which type of microscope produced them, and also when we would use the different type of microscopes. Let's talk about preparation of specimens. Mainly we're going to discuss preparations of specimens for the light microscope. This is because you're going to be dealing with light microscopes. It'd be wonderful to have an electron microscope, but we don't have one. So we're going to be talking about preparing smears for staining, simple stains, differential stains, and special stains. An awful lot of this we're going to be going over in lab as well. Let's talk about preparing smears for staining. Now a lot of this is going to be review from lab. You've done some of this already. But reviewing is good. Repetition, repetition, repetition. Alright, a smear, as you know from lab, is taking a sample and putting it out, smearing it on a glass slide. Now, as you may remember from lab, if it's a liquid sample, you just put it on the glass slide, spread it out, let it dry. If it's a solid sample, it generally works best if you mix it into a drop of water. Breaks it up, spreads it out so that you get a nice thin layer of your sample on the glass slide so that you don't have big clumps. Fixation is attaching your sample to the slide. The only fixation that you need to worry about for this class is heat fixing. That's where you pass the glass slide through the flame to denature the proteins just a little bit, enough to stick the sample to the slide so it doesn't slide off when you go and rinse your slide, your sample. Now let's talk about staining. Positive staining is where you stain the actual cell. Now most cells have a negative charge. So what charge would a positive stain have to stick to the actual cell? It would need to be a positive charge. It's part of why we call it a positive stain. Also, because it produces a positive image. Also, we call it a basic stain because, that's right, bases have a positive charge. Now for negative staining. Negative staining is where we stain everything but the cell. You're probably wondering why in the world would we do such a thing? Well, it's because when you do positive staining, the stain kills the cell. Sometimes it distorts it, so that you get little raisin cells, and they don't look like they should. So when you do negative staining, you can see the shape of the cells in your specimen without turning them into little raisins. Now, in this example, we have a combination of positive staining and negative staining. We've stained the cells on the inside, then we've applied a negative stain so that we can have a capsule show up. Now a capsule is composed of carbohydrates which normally have a negative stain, a negative charge, but in this situation they repel the stains. So your capsules aren't going to be stained by the positive stain, and the negative stain is repelled, and you get this nice spot showing you where the capsule is. Now, simple stains are where you use just one type of stain. You use a basic stain or a positive stain to stain the actual cell. Now, we use simple stains to determine cell shape and cell arrangement. Now, cell shape, sometimes we refer to that as cell morphology. Morphology is a fancy term for shape. So in this slide, if you look very closely, you'll see that the cells are either round or they are very short rods. Now, their arrangement, we see some that are in lines. Like right here, we see some that are in lines. There's some lines here. There's also some clusters, but we see an awful lot of individual cells. All of this becomes important in identification. Differential stains tell you something about your cell other than morphology and arrangement, although you can determine morphology and arrangement at the same time. Differential stains usually tell you something about the cell wall of the organism. Let's take gram stain for an example. In a gram stain, you apply one stain. We call it the primary stain. It's crystal violet. It's violet, it's purple. Then we put a mordant on. a mordant is something that helps the primary stain stay. In this case, it's iodine. Then, next, we would apply a decolorizing agent. Now, some of these cells are going to lose the primary stain. In this case, the little round guys kept the primary stain. The rod-shaped ones lost the primary stain. Now, clear against a bright background is very hard to see, so we apply a secondary stain, in this case safranin, which is red, and we get cells that are gram-positive, being purple. That means they've maintained or kept the primary stain, and gram-negative, which are those that have lost the primary stain and have been stained by the secondary stain. And here is a micrograph showing a gram stain. Now, another type of differential stain that is very similar is the acid-fast stain. Here's a picture of that. The colors have been reversed. The red cells are actually the acid-fast positive. Blue cells are acid-fast negative. In this situation, we're using different stains, and we're using an acid decolorizer. That's why we call it acid-fast, is those that are positive that hold on to the stain even though the acid decolorizer is being applied are acid-fast. It's kind of like dye-fast, where you don't lose your dye in the washing machine and everything turns out pink. So, in this case, in the next chapter, we're going to learn about why gram-positive and gram-negatives stain differently under the gram stain, and we're also going to learn why acid-fast positive organisms stain differently than acid-fast negative organisms. Now, some stains are considered special stains. They have a tendency to show us things about the cell that are more than just what the cell wall is made of. So for our first one, this is an endospore stain. In the next chapter, we're going to talk about what endospores are, but they're tiny structures that form in some types of bacteria that help preserve the cell during extreme stress. Now, they don't like to be stained, so we have to use a special staining procedure to stain the endospores, and then we stain the rest of the cell a different color. You'll be doing this in lab. This one that I showed you before, a capsule stain, is considered to be a special stain too. Shows up capsules. Now this one is a flagellar stain. Now, as we'll learn in the next chapter, flagella are hair-like extensions that help the cell move. And they're very, very thin, and they're very, very hard to see. With some high-quality light microscopes, you can see them. You can see them with phase contrast and dark field microscopes. But often what we do is we apply a flagellar stain. It's kind of like applying makeup, okay? Mascara to eyelashes. I don't know about the rest of you, but I've got skinny blonde eyelashes that you can't see. So what do? I coat them with mascara. So this is the same thing as a flagellar stain. We coat those flagella with the dye that specific flagella makes them able to be seen. That's it for this chapter. An awful lot of this chapter you're actually doing in lab, and so this will be a nice kind of a theoretical background for what we're doing in lab.